Okay, so we're going to go ahead and talk about the study guide that was given today for your term test, either on Thursday or Friday. All right, so we're going to start out talking about scientific method. So to describe the scientific process, we want to talk about what these letters mean. So OQ Hertz meant that there was observation, question, then you make your hypothesis, then experiment. From there you gather data that gives you your results, and finally you make your conclusion. Okay, a controlled experiment is any time that we are going to be using these independent and dependent variables, and obviously the big part of it is that you have a control. So a controlled experiment is when we have something to test and we have something to compare results to, and so you would always want a control. You would want constants and dependent and independent variables. Okay, how are independent and dependent variables different? Well, the independent variable is going to be something that the researcher decides to test. Researcher decides to test. And the dependent variable is going to be what changes due to the variables that the researcher set. So these are going to change because of independent variables. And we talked about in class that dependent variables are always on your y-axis, independent variables are always on the bottom. And so maybe you're testing color of eyes as your independent variable, and then your dependent variable would be the number of people that have that color eyes. So if this was brown and there was 4 people that had brown, the dependent variable is going to depend on the amount of people that have these color of eyes. All right, what's the control in the scientific experiment? This is what we compare our results to. And they're awfully important because you could do all of these tests, but if you don't have what is called like a baseline, and we don't have something to compare it to, our data wouldn't be very meaningful. So it's a baseline, and that's why it's very important to make our results meaningful. Okay, a hypothesis is a testable explanation. It must be written in the if-then format. In the space below, write a hypothesis concerning your grade on the final exam. Remember to use specific, be specific and realistic. So maybe one would be, if I study for my final, then I will pass. And then we always need those 2 supporting details, because I will remember the information that was taught in class, and I might pick up a few things that I didn't know before. So maybe I'll understand concepts I didn't in class. So hopefully everybody is studying for your final, and all of you that are listening are, so great work. All right, now we're going to actually apply this to an actual problem here. So this one, she is pouring Coke into a glass. There's always 2 to 3 inches of fizz at the top. When she is thirsty, she wants to get it down quickly. She thought of ways to reduce the foam. Once it's in the glass, so she can simply wait, she could blow on it, she could put ice on it, or she could put her finger in it. She got 4 clean glasses and a new 2-liter bottle of Coke. She poured 6 ounces of Coke in each glass and proceeded to use one method on each glass of Coke. She timed the amount of time it took for the fizz to go down in seconds by using a stopwatch. Write a hypothesis for this experiment. So we're going to have to use that if-then statement. So if, and now you could pick. So a hypothesis doesn't necessarily have to be correct. It just has to be a very strong statement. So you have to pick one. So it doesn't matter which one you pick. I guess I would probably put ice on it. So if you put ice on Pop Fizz, then it will disappear. Then give me the 2 reasons. Because, that's shorthand, ice will pop the bubbles, and maybe another reason would be that the temperature change would affect it. Okay, so you can say anything you want on these as long as you have a prediction, okay? So the independent variable in this experiment is what she's deciding on, testing, and then the dependent variable is going to be because of these. So the independent variables are going to be the ways that she can reduce foam, so ways to reduce fizz or foam, either way. And then the dependent variable is going to be the time it takes her to do so. So if we were going to graph this, we could put as our independent variable, we could put weight, which would be our control in this experiment, because that's something normal that you can compare your results to. She could blow on it, and she could put ice in it, and then I'm out of room. And then now the amount of time that it takes, we can make a little bar graph, and the time is going to depend on which thing she does. Okay? So, that would be a controlled experiment that you could do, and how you would write the hypothesis for it. Okay, now this would be moving into chapter 2. Atoms are composed of 3 subatomic particles. So 3 particles are protons that have a positive charge, and they are in the nucleus. Then we have neutrons that are neutral, and they are also in the nucleus. And then we have electrons that are negative, that are in the shells, or you could say cloud if you want it, not cloud. And we can draw these for an atom of nitrogen. So we can have nitrogen has this number right here, tells us the number of protons and the number of electrons. They're always equal when you're drawing an atom. So if I drew 7 protons, and then we would have 7 neutrons, because 14 mass number minus the atomic number equals the number of neutrons. And so 14 minus 7 gives us 7. So 1, 2, 3, 4, 5, 6, 7. And then we're going to have 7 electrons. Remember, there's only 2 on the innermost shell, and so then we need 5 more. 5 on the outermost shell. Okay, the atomic number tells us the number of protons and electrons. And why atoms are considered neutral is because protons are equal to the number of electrons, and so that cancels out the positive and the negative charge, which will always be neutral. Okay, but when they're not neutral, that is when we have an ion, and this is, an ion is when we give or take electrons. So if you have an element like sodium, who has 1 on its valence shell, because he's in group 1, that's right, its symbol with his valence shell. And you have chlorine over here, he has 7 in his valence shell. Sodium can give 1 to a chlorine, this making, when he loses an electron, he's going to have more protons than electrons, so he's now going to be a positive ion. And then chlorine's going to gain an electron, which makes him have a negative charge. And then they're going to be like a magnet, and they're going to form an ionic bond, because one's positive and one's negative. You become positive when you lose electrons, and you become negative when you gain electrons. Describe a covalent bond. I'll give examples and draw a diagram illustrating covalent bond. So covalent bond is sharing of electrons. And you could show that something was being shared, like in water, for instance. So if we had oxygen here, it has 6 valence electrons. Okay, and then we could have hydrogen sharing that electron right there and sharing this electron right here. And hydrogen has 1 electron, so now these electrons are not stationary, they're moving all over. And so now this hydrogen has 2 orbiting around it, and this electron here is going to actually orbit around oxygen too, so they're sharing electrons. Okay, ionic bond is going to be made of ions. Okay, so you can refer back up to this guy there. It's made of ions, it gives and takes, and if we're going to diagram it, it would be right there. Okay, chemical reaction has occurred. Well, if you're looking at just the equation here, the arrow is the indication that something has been yielded, but also when the atoms are reorganized or rearranged. Okay, so as you can see down here, we start out with CO2 and H2O, and we have the same atoms. We have CO, and we also have H and O, but now they've been rearranged, and so when they're reorganized or rearranged, we know that a chemical reaction has occurred. We want to identify the products, so whatever's on at the beginning is always the reactants, and whatever is produced is the products. Alright, endothermic and exothermic. These were the demonstrations that we did in class. So endothermic, and we just learned this prefix. We saw it again with the cell membrane. So endothermic means energy or heat is absorbed, goes in. So that's when the chemical reaction was done. It was cold because it was stealing the heat from the glass container. And then the exothermic reaction is heat released. Okay. Okay, so endothermic and exothermic reaction, that is when we have our 2 charts here. And as you're going up, this is the amount of heat, the change in heat. And we always start out in that beginning little arrow, or the beginning of the graph is our reactants. And as we go up, this is the amount of activation energy that will be needed in order for a chemical reaction to take place. And at this very peak, that is where the chemical reaction is taking place. And then as we move down, this is where our atoms are being rearranged. And then when we finally get to the bottom, this is going to tell us which one is endothermic and which one is exothermic. And our exothermic reaction is going to be lower than our starting, because there's going to be a heat produced or extra energy released, and so it's going to be lower because this is the amount of energy along this y-axis, whereas the endothermic is going to go up. Here's the chemical reaction takes place, and we're going to have energy that is absorbed. Okay, so endothermic is always higher from its starting point, and exothermic is always lower than its starting point. Okay, and so activation energy is going to be this here in the middle, how long it took to get to the top, and reactants are always at the beginning, and products are always at the end. Okay. And this is, I'll label it real quick. This is our exothermic. This is our endothermic. Okay, pH scale. This is sometimes opposite than what we think. 7 is always neutral. And as we go higher, up to 12 or 14, this is going to be your base. And when we go here, this is going to be our acid. 1, and acids have a very high concentration of hydrogen ions, and bases have a very high concentration of hydroxide ions, or you could also say bases have a low concentration of hydrogen ions. Okay, 2 unknown substances, and you are testing their pH. If we have something that's 4.56 and 411, which substance is a stronger acid? So as you go lower, it's more acidic, so the lower number of these two are going to be a stronger acid. So, and we know that because the number is lower. Okay, your notes on page 54 in your textbook is about an enzyme reaction. And what we really want to talk about is labeling this. So in your book we have a little enzyme that maybe looks like this, and you'll see these drawn a variety of different ways, and this is the actual enzyme, and this is where our chemical reaction is going to take place, is in here, and then we call this the activation site. And then we have our substrates that are also called our reactants. And in this case, we'll have 2. And then they both go into our enzyme. And they fit like a lock and key, and this is where the chemical reaction takes place, and when it is done you will have a new molecule. So you got to think of these as more of a molecule than triangles, but it's a good way to illustrate it. So these are going to be your products that now are bonded together instead of being apart. Okay. All right. So here's a little matching with our monosaccharides. So a polymer of a nucleotide. So that's going to be a whole bunch of nucleotides put together, and so that is going to be DNA. A monomer of a carbohydrate. That's going to be a monosaccharide. 2 monosaccharides chemically combined together. Example, sugar. We saw this in our lab, and that is a disaccharide. Joined together with a peptide bond. Peptide bond is what links amino acids together, and so that would be a protein, which is right here. Composed of glycerol and 3 fatty acids. 3 fatty acids is a triglyceride. That's E. Controls rates of chemical reaction and builds muscle. Builds muscle. That is protein. Chemical reaction that reactions and builds muscle. So that is going to be, this is supposed to be amino acids. So it says join together with peptides, and I even said amino acids, but then I wrote protein. Sorry about that. Control, so this one's going to be F, so it should be B, D, F, A, C, E. Okay, and then many monosaccharides together. That's a polysaccharide. Carbon molecule that stores genetic information. It's going to be a nucleic acid. Carbon molecule. Main component of the cell membrane is phospholipid. Stores energy, that is a lipid. Quick energy, that's carbohydrate. And monomer of a nucleic acid is a nucleotide. Okay, be able to recite the three components of the cell theory. Okay, and in my class we did BEAM, and that is that the cell theory says it's the basic unit of life. Every living thing is made of cells. And then our last one was mitosis, which just means cells are made from pre-existing cells. Okay? Okay, so the next thing here is how can you tell if these cell is eukaryotic or prokaryotic? So eukaryotic cells are going to be animal and plant cells. And prokaryotic cells are going to be bacteria. And remember, bacteria is going to have no membrane-bound, membrane meaning that it's enclosed, membrane-bound nucleus. It has free-floating DNA everywhere. DNA, no membrane-bound nucleus, that's what I was spelling. And animal and plant cells are going to be much bigger and more specialized. And they have more organelles, where bacteria they're pretty small and they have very few organelles. All right, and what three organelles are found only in plant cells? Okay, the cell wall is obviously the big one. And then we have the chloroplast. And then we have that big central vacuole. It needs lots of water. So it has that big central vacuole inside of it. Chloroplast, that's where photosynthesis takes place, and the cell wall gives it structure. All right, so here are the organelles. So you need to know the functions. So vacuoles, they are going to store water, and they're also going to store other materials if needed, but water and other materials. Stores, that's probably important, right? Stores water and other materials. And they kind of just have holes in the top. They kind of look like this, and the big central vacuole is like really large and it's indented in the plant cell. Mitochondria, this is where cellular respiration takes place, and this is where oxygen is turned into carbon dioxide and energy is produced. So energy production. And we talked a little bit about ATP with active transport, and this is where ATP comes from, is made. And he looks like this, and he has a whole bunch of surface area for lots of chemical reactions to take place. The Golgi apparatus is kind of looks like a whole bunch of circles squished together. So I like to draw it as circles squished together. And what he does, it's actually kind of circles. There's a whole bunch of membranes stacked together, and he deals with transport and he does processing and packaging. We'll put packaging. He's kind of like the UPS guy, right? For proteins, transport, packaging, and shipment. Ships proteins. All right, cell membrane. I think you guys definitely know this one now. Has the phospholipids. We have some proteins. And the cell membrane is for it gauges the material in and out of the cell. The rough ER, this is protein production and transport. And this is right around the outside of the nucleus. So it usually has a kind of circular shape, and it has little dots all over it, little ribosomes. Okay. Smooth ER is for transporting proteins. Proteins is a big function of our cell, obviously, and it's going to look similar to your rough ER, except for it has no dots because it does not have ribosomes on it. All right, the centrioles, those are like little licorice cylinders. They have lots of lines on them like this. And they're found in the cell. And later on, we're going to learn that they help in cell division. Oops, that's supposed to be over there. Cell division. And spindle fibers that help break apart that cell. Okay, lysosome, these have digestive enzymes. And they house the chemical reactions in the cell, and they're going to be little similar looking to a vacuole. They have like a smaller head with a more like larger body. Okay, the ribosome, this is makes proteins from mRNA, and they are little tiny dots for the most part. Okay, nucleus, this is storage of DNA and the place where RNA is made. Okay, and the nucleus is a circle with a little nucleosome inside, and that's where all the DNA is in its chromosomes. Nucleolus, we can draw that a little bigger here. And this is where chromosomes are stored. And then the cytoskeleton is for structure, and it also is used for transport throughout the cell. That's little microtubules and microfilaments. Those are important, and they kind of have underneath the lipid bilayer or the cell membrane, they kind of give the structure. Cytoplasm, this is the jelly-like substance that is inside the cell that holds the organelle. I guess I don't know how to draw it other than having little organelles inside. Okay, and then the vesicle is the last thing, and this is just a small membrane sac, and it holds and recycles. It helps with storage and transport of proteins, and so it's kind of like it's another little thing that has a hole in the top. They all kind of look similar. These are a little smaller than the lysosomes. and they are the recyclers used for recycling. Okay, intracellular receptor and membrane receptor. How are they similar? So if you go to page 80 in your book, there's two really good pictures of these. So your intracellular receptor is going to just mean intra, meaning inside of the cell membrane, and there's going to be something that passes across, the cell membrane, and there is a receptor on the other side that fits into it. And once it's that lock and key fit, then it spurs something to happen inside the cell. And then we actually have membrane receptors that are inside of the membrane, and so they have a similar look here, and so something comes in and attaches, and then this makes something else come out on the other side. So maybe it has, like this, and as soon as that attaches, then this can leave and sends a message or a variety of different things. But the point is that membrane receptor is on the cell membrane, where the other one is on the inside of the cell. Okay, passive and active transport. So passive transport has no energy needed, okay? And there's three different types. We have diffusion, osmosis, and facilitated diffusion. Diffusion is dealing with gas or liquid, where osmosis is dealing with water movement in and out of the cell. And then facilitated diffusion uses a protein channel, but none of them require energy. Then we have active transport, and this requires energy. And this is generally ATP. And you can have active transport with protein channels. And we have endocytosis. And endocytosis, there are two types. There are the pino for liquids, pinocytosis, that deals with liquids, and the phagocytosis that deals with solids, typically foods. And then we have exocytosis, and all of these take energy in order for it to happen in the cell. And exocytosis is when something is going out of the cell. Endocytosis is when something is going in. All right, describe the structure of the cell membrane. Okay, so we're going to have our phospholipids that have our glycerol and our two fatty acids. Okay. Then we're going to have big protein channels, and we have all sorts of specialized proteins. I think all of the teachers teach this a little differently. I mean, some of us go into more depth about the proteins, so we'll just be kind of general. So the lipids that we're talking about are the phospholipids here. Also, there's some lipids. We learned earlier that cholesterol is a type of lipid, and it's actually used as support through these fatty acids for extra support for structure. When it gets cold and hot, it holds the fatty acid tails together. Okay, so if this is the inside of the cell, we would have cytoskeleton right inside here for structure. And then carbohydrate chains come up out of those proteins that are right here. So we have carbohydrates that are like little chains, and they can even be out of the phospholipids, and then proteins. And we have all sorts of different types of proteins that you can have in the cell membrane as well. Okay, prokaryotic and eukaryotic. I think we already talked about that one earlier. But I think this one says similarities. Well, they're obviously both cells. They both have DNA. They both have mitochondria. And there's probably a couple of other similarities, but I think if you knew those three, that would be enough. All right, and then be able to describe the effects of the cell while placed in isotonic, hypertonic, and hypertonic solutions. And you can draw diagrams to help you understand this a little better. So when you have an isotonic solution, okay, that means the solution around it, we have a cell, everything is going to be equal. That means they're both kind of like a medium concentration, and there's the same concentration on the inside and the outside. Okay? Hypertonic solutions mean that there is going to be a high concentration of solutes on the outside of the cell. So if the cell's in here, there's a high concentration. Well, I shouldn't have drew my cell yet. So what this means is the water moves out of the cell to try to even out, and it makes this shriveled cell in a hypertonic solution. So hyper means the cell shrinks, cell gets smaller. And then the last one, hypotonic solution, means that there's lots of solutes on the inside of the cell, and so water goes into the cell and makes it really big in comparison to what it started with. Maybe it was, started out being small. Once all the water moves in, and it gets larger. Okay, and that's also on page 82 of your book if you would like to look at those. All right, I hope this helped, and good luck on your